this video, we are going to learn Four-Klein method to find genotypic ratios among progeny in these two crosses. Actually, this is a requested video by Nati Arnold. These two crosses might be imaginary ones written just for practicing Four-Klein method. So let's assume that these are the genotypes of an organism, and also suppose that these three genes are assorted independently. As we are studying three genes here, it is a trihybrid cross, and the alleles of these three genes are capital A small a, capital T small t, and capital X small x. So let's find out the genotypic ratios of this first cross. This is the first cross. In Four-Klein method, we usually consider each gene separately in a monohybrid cross and find its genotypic ratio. This is easier than considering all the three genes simultaneously. So let's take the first one. These are the parents. These parents have heterozygous genotype. In heterozygous genotype, while formation of gametes, half of the gametes receive dominant allele and the other 50% gametes receive recessive allele. So two types of gametes are formed. Here also, two types of gametes are formed because the parent is heterozygous in condition. When these gametes fuse, the progeny are formed. If these two gametes with dominant alleles fuse, an organism with homozygous dominant genotype is formed. The fusion of a gamete with dominant allele and a recessive allele leads to the formation of a heterozygous organism. Likewise, the fusion of this gamete with this gamete leads to the formation of an organism with heterozygous genotype. The fusion of gametes with recessive alleles leads to the formation of an offspring with homozygous recessive genotype. So these are the possible genotypes of progeny in this cross. Let's look at the ratio. The genotypic ratio of these offsprings is 1:2:1. This ratio can also be represented in the form of fractions like this. Here I have written the genotypes horizontally. But while writing the Four-Klein method, I will write the same genotypes vertically like this, along with their ratios. We will consider these genotypes and their respective ratios of monohybrid cross and write the Four-Klein method. Capital A, capital A, capital A small a, and small a small a. These three genotypes of offsprings we got from monohybrid cross previously. Now let's consider the second cross. Let's consider the second gene and write a monohybrid cross for that. We'll get three possible genotypes for that. Let's see it. Capital T small t into capital T small t. As this is heterozygous in condition, it also produces two types of gametes here. This organism also produces two types of gametes. The fusion of these gametes leads to the production of progeny. When these two gametes fuse, a homozygous dominant genotype is formed. When the gametes with capital T and small t fuse, a heterozygous genotype is formed. Same thing here. Likewise, a homozygous recessive genotype is formed due to fusion of gametes with recessive alleles. So the genotypic ratio here is 1:2:1. Let's consider this monohybrid cross genotypic ratio and write it in Four-Klein method. Capital T, capital T, capital T small t and small t in the ratio of 1:2:1. This means if a progeny inherits capital A in its genotype, it may have a combination with capital T or these two. We are writing the possible genotypes here. The same combinations may also go with capital A small a. Now the combinations with small a small a. Till now, we saw monohybrid crosses for the first two genes. Now let's consider the third gene, which is capital X into small x. So let's write monohybrid cross. This is heterozygous in condition, so it produces two types of gametes, dominant allele and another gamete with recessive allele. This is homozygous recessive in condition, so it produces only one gamete which has recessive allele. The fusion of these two gametes leads to the formation of heterozygous genotype, and these two gametes fuse to form an organism with homozygous genotype. So here the genotypic ratio is 1:1. That means half is to half. Let's consider this monohybrid cross genotypic ratio and write in the fork-line method here. Capital X, small x in the ratio of 1:1. This means an organism with capital A, capital T genotype may have a combination of capital X or small x. Let's write the possible combinations for this also. If you want to follow through all these genotypes clearly, please write down this cross on a paper along with me. So till now we have seen the genotypes separately. Let's combine them together to look at the actual possible genotypes of offsprings. Capital A, capital T, and capital X, small x is the actual possible genotype of offspring for this cross. What is the probability of occurrence of this genotype? To get that, we have to multiply their individual probabilities. Why is it so? Because in mathematics you would have studied the multiplication rule in probability. According to this, when three independent events occur simultaneously, their probability is equal to the multiplication of their individual probabilities, which is 1/4 into 1/4 into half. These are their individual probabilities. So when these three independent events are occurring simultaneously, their probability is 1/32. Why do we consider these three as independent events? Because these three genes are assorted independently. So the same goes with the remaining genotypes also. Let's look at the next one. Capital A, capital T, and small x occurs at a probability of 1/32. The next one, capital A, capital T, small t, and capital X, small x occurs at a probability of 2/32. Because this probability has changed here. The next one, capital A, capital T, small x, 2/32. That next one. This first genotype occurs at the probability of 1/32, which is capital A, capital T, and capital X, small x. 1. The next one is this, and so on. The next genotype, capital A, small a, capital T, capital X, occurs at a probability of 2/32. That is this one. And the next one, and so on, till here. And this one, small a combinations, small a, capital T, capital X, small x occurs at a probability of 1 by 32. The genotypic ratio among progeny of this cross is this. This is the second question. In this question also, let's consider only one gene first and write the monohybrid cross of this. We have already watched this for the first question. Parents are heterozygous, so two types of gametes are formed, and these are the progeny. The genotypic ratio is 1:2:1, which is 1/4:2/4:1/4. Let's write the Folkline method for this cross. So we got capital A, small a and small a in the ratio of 1:2:1. Now we should consider the second gene, which is T, but here it is heterozygous in condition and this is homozygous in condition. So we'll get only two possible genotypes. Let's see how. It's monohybrid cross. This heterozygous genotype produces only two types of gametes and this one produces one type of gamete. The fusion of these two gametes produces homozygous dominant genotype and these two gametes fuse to produce a heterozygous genotype. Here the genotypic ratio is 1:1. We consider this genotypic ratio of the monohybrid cross and write it in Folkline method, which is capital T is to capital T small t in the ratio of 1:1. That means an organism with capital A genotype may have a combination of capital T or may have a combination of capital T small t. The same goes with capital A small a. Next one, small a. Now we have written the possible genotypes of first two genes. Let's consider the third gene and write the monohybrid cross for that. Here we'll get capital X and small x. We already watched this monohybrid cross for the first question. Anyway, let's do it again. Capital X into small x as given in the question. So we got a genotypic ratio of 1:1, which we considered in writing the Folkline method. So let's write all the combinations. This is the actual possible genotype of the offspring, where we have combined the three separated genotypes, capital A, capital T, and capital X, small x. Now the probability of this genotype is 1/4 into half, which is 1 by 16. Notice here the probability is changed because the genotypes of the parents have changed. Earlier you have seen that capital T small t is crossed with capital T small t, but here it is crossed with a homozygous dominant. That's why the probabilities have changed here. Next one, capital A, capital T, small x, and here combinations with capital A, small a. Next combinations with small a. So these are the possible genotypes and their probabilities of progeny formed in this cross. Let's write the genotypic ratio properly. Capital A, capital T, and capital X, small x occurs at the probability of 1 by 16, 1. You can see here 1:1, which we got here. Let's move this one. The next one, because it is heterozygous here, capital A, small a, combinations are 2:2. Next one, small a combinations are 1:1:1:1:1. So this is the genotypic ratio of progeny born in this cross. If you want to learn this method easily, practice first in a dihybrid cross with some real example. That way you can learn both phenotypic and genotypic ratios.